Good day everyone, we will be presenting an important cloning vector in molecular biology and genetic engineering. It is a phage, it is a plasmid, it got the best of both worlds, introducing Cosmids. Here is a quick overview of our presentation. We will be reviewing first what are cloning vectors, the role of plasmids and Lambda phage as cloning vectors, and finally the composition and uses of Cosmid vectors in different fields of science.
But before we discuss about Cosmids, let's have a review first on cloning vectors and their features. So what is a cloning vector? Cloning vectors are DNA molecules into which foreign DNA can be inserted. It is a small DNA molecule that is capable of self-replication inside the host cell. Furthermore, cloning vectors are used as tools to transfer a foreign DNA fragment of interest to the host cell. Now that we have already defined what a cloning vector is, let's take a closer look at some of the features that all cloning vectors require. The first feature is the origin of replication, or the ORI. It is the sequence at which replication of DNA begins. This sequence is also linked to the copy number of the vector and controls how many times your gene of interest will be produced in the host cell. The second feature are the selectable markers. Selectable markers are genes that help identify bacteria that have successfully transformed or taken up their recombinant plasmid. Antibiotic resistance genes are one of the most commonly used selectable markers. The last feature that all cloning vectors require are the cloning sites. All cloning vectors need sites that will allow the insertion of foreign DNA into it. Most commonly, this involves the use of restriction enzyme recognition sites.
For a review, I will briefly discuss about the common vectors used in microbiology.
Plasmids and lambda phages are the two most used cloning vectors. Let's start with plasmids. Plasmids are self-replicating circular double-stranded DNA structures. It is utilized as a vector in bacteria, yeast, and higher eukaryotic cells. A gene of interest must be put into a plasmid, and the plasmid and the gene must be using a restriction enzyme. The recombinant DNA is then transformed into bacteria and allowed to proliferate before being subjected to further manipulations. The lambda phage, a bacteriophage that infects E. coli, is a temperate virus capable of residing within its host genome via lysogeny. Phage vectors are quite diverse and can be divided into two groups: replacement vectors, which are useful for cloning large DNA fragments and are utilized in the building of higher eukaryotic genomic libraries, and the insertion vector, which is employed in cloning tiny DNA fragments such as cDNA.
So in the third part of this presentation, I will briefly discuss about the structure of Cosmids, including a sneak peek on the steps in DNA cloning using Cosmids. Cosmids are medium-sized cloning vectors. They are basically cos sites plus plasmids, hence the term Cosmids. Cosmids are hybrid plasmids developed by combining the features of a plasmid vector and a bacteriophage vector. The cloning capacity of these vectors is 35 up to 45 kilobases. They may also have one or two cos sites present in their lambda phage cos sequence. If they have suitable origin of replication, then they can replicate as plasmid within the host cell. The following comprise the basic features of a cosmid. First, the cos site, which provides blunt ends. Next, the recombinant site, indicated by selectable markers such as the antibiotic gene in the figure. Next, the ORI-V, or the origin of replication. And lastly, restriction endonuclease sequences, which is represented by EcoRI and SMA1 in the figure. DNA cloning using a cosmid typically observes the following steps. First, the ligation of a foreign DNA between two cos sites. Second, a concatenaric DNA is made. Third, DNA is introduced into phage head using in vitro packaging to form matured phage particles. Fourth, introduction of the cloned DNA into E. coli by transduction. And fifth, after their entry into the host cell, the cosmids are maintained as plasmid.
Now let's take a look at some commonly used cosmids. Cosmid PJB8 is constructed from E. coli plasmid PBR322 and two cos ends of lambda phage DNA. It is 5.4 kilo base pair in size, contains ampicillin resistance gene, and restriction endonuclease enzymes EcoR1, BAMH1, and HinB3. Similarly, Cosmid PLFR5 is derived from E. coli plasmid PBR322 with 2 cos ends from Lambda phage. It is slightly larger with 6 kilo base pairs, and instead of ampicillin, it contains tetracycline resistance gene. There is also a multiple cloning site wherein the gene of interest may be inserted. Cosmid PHC79 is 6.5 kilo base pairs in size. It can carry relatively large DNA fragments and contains both ampicillin and tetracycline resistance genes. Foreign DNA can be inserted into EcoR1, BAMH1, or HindH1 restriction sites of the Cosmids. Lastly, SuperCos1 is 7.9 kilo base pairs, which can carry DNA fragments from 30 to 50 kbp. It contains 2 marker genes for ampicillin and neomycin resistance, T3 and T7 promoter regions, and not one restriction site. The presence of promoter sequences can direct cell-specific expression, which is useful in eukaryotic cells.
Cosmids are highly versatile. In fact, it has a wide range of applications. In the next slides, we will be discussing on how cosmids are exploited in different fields such as agriculture, medicine, genomic library, and gene expression. Pea toothzeri contains genes encoding nitrogenase. On the other hand, pea protogens do not possess this. Cosmids can be exploited in agriculture. One example is the study conducted by Seton and colleagues last 2013, whereas they isolated 255 base pair fragments from pea toothzeri. X940, a recombinant cosmid containing NIT genes, was sequenced by primer walking. X940 was introduced through transformation into competent cells of different Pseudomonas species. Genomic walking assay and southern blot was performed to monitor the X940 cosmid insertion within pea protogens PF5. Recombinant bacteria PF5 X940 exhibit nitrogenase activity and high ammonium production. Plant species inoculated with PF5 X940 have increased productivity under low nitrogen soil.
Cosmids are also applied as cloning vectors in medicine. One example is the overlapping cosmid clones method. An advantage of this method is that it does not require plaque purification because all reconstituted viruses are recombinant, allowing this technique to be widely used. This method has been utilized to generate a large number of mutants, especially for equine herpes virus (EHV), Marek's disease virus (MDV), and turkey herpes virus (HVT) to study gene function. In 2002, a recombinant Marek's disease virus, or MDV, was successfully generated using overlapping cosmid clones. In the study, 5 overlapping MDV DNA fragments were generated from the VV-MD5 strain of MDV after digesting with different restriction enzymes, which were cloned into cosmid vectors, generating overlapping cosmid clones named SN5, P89, SN16, A6, and B40. The MDV PP38 gene located in cosmid A6 was then deleted using a REC-A-assisted restriction endonuclease or RARE cleavage method. Finally, unmodified cosmids and mutated cosmid A6 were digested and the 5 overlapping viral DNA fragments were purified and co-transfected into cells to produce the recombinant MDV with deleted PP38 gene. This recombinant virus produced showed that PP38 is important for MDV early cytolytic infection.
Cosmid vectors are tools in constructing genomic libraries. So what are genomic libraries? According to Burks, it is a collection of the total genomic DNA from a single organism created by isolating DNA from cells and then amplifying it using DNA cloning technology. The DNA is stored in a population of identical vectors, each containing a different insert of DNA. Cosmid libraries can represent an entire genome in a library of circular DNA molecules, allowing for the faithful amplification, cloning, and isolation of large genomic DNA fragments. In a study by Iqbal and colleagues, environmental DNA was extracted from soil samples and were ligated with either cosmid vectors PJWC1 or PJSS in the SCA1 cloning site. They were introduced in E. coli EPI 300 by phage transduction. The infected bacteriolon was plated on a lysogeny broth or LB tetracycline plate to estimate the size of the library. The number of colonies on the titer plate was counted to estimate the number of clones. In total, 500,000 and 200,000 unique Cosmid clones were constructed using vectors PJWC1 and PJSS respectively, constituting 22.5 gigabase of eDNA. Subsequently, DNA prepared from these libraries were electroporated into R metallidurans and screened for antibiosis using the top agar overlay method. The researchers revealed that 6 Cosmid clones in R metallidurans contain several lipase, proteases, and cell wall lytic enzymes as seen in a zone of inhibition around individual colonies.
Now let's discuss about the involvement of Cosmids in gene expression. In a study by Braña et al. In 2014, Streptomyces albus and Streptomyces lividans were transformed with Cosmids containing the THN gene cluster from Streptomyces cattleya. The Cosmids used were COSCAT25, COSCAT14, and COSCAT22, which contain different regions of the THN and CPH2 gene clusters from S. cattleya. Furthermore, Cosmid COSCAT32 was also used since it harbors the complete nucleotide sequence. In the results, phenotypic changes were detected in S. albus and S. lividans. This prompted further analysis involving comparative metabolite profiling. Metabolite profiling of the recombinant strains showed differences in multiple metabolites, which involved activation of at least 6 silent metabolites and silencing of at least 2 active metabolites.